Hey everybody, we are back here in the lab today and we are going to be working on our second set of selective and differential media today. So the first set we did were SBA, starch, gelatin, and MSA, and today we're going to do the second half of the selective differential media, which is PEA, McConkey, EMB, and HEA. So those are the last four that we're going to do today. So I'm going to go over the background information on those four. We're going to inoculate those four types of media. So to start out today, we are going to start with PEA. And in my mind, I like to separate these types of tests depending on what they're testing for and what kind of media it is. It makes it easier for me to remember. So when I group them, I group PEA all by itself because it's completely different than everything else. And then I do the last three, which are McConkey, EMB, and HEA, because those three are all similar. They're a little bit different, but they're very similar to one another. So we're going to start out with PEA. And PEA is the only kind of media out of these all 8 types of media or tests that is strictly selective. So it doesn't really tell you a whole lot of information. So it's just strictly selective. And so what PEA stands for, so I have it up there, right up here. What PEA stands for is phenyl ethanol agar. Okay. Now in your book, I think it's phenyl ethyl alcohol, but ethyl alcohol is the same thing as ethanol. So we just shorten it. So phenyl ethanol agar, that's what the PEA stands for, and that is the selective agent. So the phenyl ethanol is the substrate and the selective agent. It's the only thing that's in the media. Okay, the main substrate that's in the media is phenyl ethanol. So it's a type of alcohol. So PEA is going to select for gram-positive organisms. It selects for them. So that means that only gram-positive organisms are going to be able to grow on PEA agar. So if you think that your unknown or whatever kind of bacteria that you're dealing with is a potential gram-positive organism, when you streak it on PEA, it should be able to grow. So it selects against gram-negatives. Gram-negatives are not going to be able to grow on PEA. And the reason why is because of the selective agent, which is alcohol, phenyl ethanol. So if you think back to the gram stain, remember what alcohol does to gram-negative cells. As soon as you hit those cells with alcohol, the LPS layer disintegrates, okay, and the cell wall, you create these very, very large holes in the cell wall. So the alcohol essentially kills the bacteria, so it's not able to grow. So that's why gram-negatives are not going to be able to grow on PEA. So for a test, a positive result for PEA is growth, and a negative result is no growth. And that's all that you need to know for PEA. Because it's just strictly selective, that's the only thing you need to know. If it's growth, that means it's a gram-positive organism. If there's no growth, then that means it's a gram-negative organism. So today, whenever I inoculate my PEA, I have a gram-positive cell, which is bacillus, and I have a gram-negative cell, which is E. coli. So I will streak both of those on my PEA and see which one grows. Of course, the bacillus is the one that's supposed to grow. The E. coli is the one that's not going to grow. So that's all you need to know for PEA. Now, the next 3 types of media are very complicated, I'm going to warn you. There's a lot of information that goes along with it. They're all very similar, but yet very different at the same time. All 3 of these next types of media are both selective and differential, just like the MSA. So you're going to have a selective agent in the media, and you're going to have an indicator. So not only is it going to select for certain bacteria, but you're also going to be able to see differences among the metabolism of the bacteria. So the first one that we're going to go over is McConkey. MAC stands for McConkey agar. And the substrate in this media is lactose, so it's a sugar. Lactose is a disaccharide. The selective agent is biosalts and crystal violet. So the CV stands for crystal violet. So that's the selective agent. So let's talk about that part of the media. McConkey is selecting for gram-negative organisms. Selecting for. So that means that only gram-negative organisms are going to be able to grow on MacConkey. That's the target group of bacteria that we're looking at for MacConkey when we're looking at the pathogens. We're looking for gram-negative organisms. Remember, for MSA, we were looking for a gram-positive organism. We were looking for staph. Here, we're not looking for that anymore. We're looking for gram-negatives. So we don't care about gram-positives at this point, so we don't want them to grow. So the bile salts and the crystal violet prevents the gram-positives from growing. So only gram-negative organisms are going to grow on MacConkey. That's the selective part of it. So just like the MSA, this is a two-part deal. So if the bacteria grows on MacConkey, that tells you it is some type of gram-negative organism. Then we have to look at the differential part of it. The indicator that's in MacConkey agar is called neutral red. So we've got more red color. So please don't get this confused with phenol red. And then next week we're going to go over another test that has another kind of pH indicator in it. So you're going to see that there are lots of colors, lots of indicators that all change colors. Some of them the same color, some of them different colors. So these charts are really going to help you whenever you're memorizing and trying to remember all of this stuff because it's a lot of stuff to memorize. So MacConkey agar, my indicator is neutral red. Now my substrate is lactose. So the metabolism part that I'm looking at is fermentation. So does the bacteria that's growing on the plate ferment the lactose, yes or no? So let's talk about what kind of products are going to be produced if fermentation occurs. If fermentation occurs, the bacteria is going to take the lactose and turn it into acid. So it takes the lactose, puts it through, breaks it down, puts that through glycolysis. Then right after glycolysis, the pyruvic acid is turned into acid, okay? A different form of acid. So the pH in the plate is going to go down if fermentation occurs. If fermentation does not occur and the bacteria uses the lactose for respiration, then you're going to get water as the end product, okay? Because that's the end product in respiration. You get water as your end product. That is a neutral or basic end product. So we have fermentation, which is acidic end products. We have respiration, that is neutral or basic end products. So to be able to test to see which one's happening, we need to have our pH indicator. So remember in MSA, our indicator was phenol red. Again, we were looking for fermentation. Here, our indicator is neutral red. Neutral red under acidic conditions turns, in your book it says red, but if you notice on the board, I have hot pink. The colonies are not, and when you look at them in person or on the video, whenever I show you, they do not look red, they look hot pink. And it's the colonies themselves. Now the agar ends up turning a pinkish color too, but it's the colonies that you're going to be looking at. Remember in MSA, it was the color of the agar that we were looking at. In MacConkey, you're going to be looking at the colonies themselves. So those colonies are going to turn bright, bright pink if it's positive for fermentation. So, number one, if it grew, that means that it is some type of gram-negative organism. So, okay, that's one check. That's one box that we have checked. Then, if it grew, you have to ask yourself, is it a fermenter or not? So, if the colonies turn hot pink, then that means yes. They grew, so it's a gram-negative organism, and they turned hot pink, so it is a fermenter. If you have growth, but your colonies are colorless, so they're going to be the same color as the media. If they're colorless, this is a negative reaction for MacConkey. So those bacteria did not ferment. My two bacteria that I'm going to use for my MacConkey agar are E. coli, because that's usually the bacteria that we're looking for. And then Pseudomonas. Pseudomonas is a gram-negative organism, so it is going to grow, but it is a non-fermenter. So it can't ferment the lactose. So that's going to be our 2 bacteria that we're going to use for McConkey. Now, moving on to the next media is EMB. EMB stands for eosin methylene blue. And again, for this media, we're looking for specifically E. coli. That's the bacteria that we are interested in. And for EMB, just like McConkey, the substrate is lactose. We're going to be looking at lactose fermenters versus non-lactose fermenters again. The selective agent in eosin methylene blue is eosin methylene blue. That's what the media is named for. Okay, these are dyes. They are also going to be preventing the growth of gram-positive organisms because just like in McConkey, we're not worried about gram-positive organisms here. We're looking for E. coli, which is a gram-negative organism. So if the bacteria grows, then that means it is a gram-negative organism. And the dyes here, eosin and methylene blue, are what prevents the growth of gram-positives. Now, just like the McConkey, we're looking for fermenters. The indicator in eosin methylene blue is eosin methylene blue. So the selective agent and the indicator are the same in this media. So if you get growth, number 1, that's a gram-negative organism. Then, if you get growth, we need to see what color are the colonies. Did that bacteria ferment the lactose, yes or no? So a positive result for EMB are purple colonies. Okay? Now, that purple can range in whether it's a deep dark purple or a bright lavender color purple, but either way, purple is positive for EMB. That means that this bacteria fermented the lactose. For a negative result for EMB, it's again colorless colonies. So basically the same color as the media. Now, there is something special about EMB. E. coli is the only organism that is going to give you what is called a green sheen on top of the purple colonies. So when you streak E. coli on EMB, it's going to grow. The colonies are going to be purple. And on top of the purple colonies, you are going to see a bright green sheen, and it's shiny. It is metallic. And E. coli is the only organism that does this. So if you think that whatever bacteria you're dealing with is E. coli, then streak it on EMB, and if you get purple colonies and you get a green sheen, then 9 times out of 10, that is going to be E. coli. Now, there are other tests that you can do on top of the EMB, like MVIX, that's going to further identify E. coli for you, and we are going to go over those. But EMB, if you streak it on EMB and it's purple and it's got a green sheen, it is going to probably be E. coli, unless there was something with the wrong with the media or something like that, because that's what the EMB is used for. So for EMB today, we have 3 bacteria. E. coli is going to give me the green sheen. Enterobacter orogenes is a gram-negative fermenter, so it's going to grow and it's going to give me the purple colonies without the green sheen. And then Pseudomonas is our gram-negative non-fermenter. So it's going to grow, but it's going to give me colorless colonies. So that's my 3 bacteria that I'm going to use for EMB. So if you're not confused yet, let's move on to the last media, which is HEA. So if it could get any more complicated, it is a little bit more complicated than the others that we have went over, because it's got a secondary substrate. So it's got lots of stuff in this media, so there's a lot for us to go over. So HEA stands for hectoic enteric agar. It's kind of a crazy name, but we are testing for enteric bacteria, so that's where the name comes from. And the substrate here is lactose. So just like with McConkey and EMB, we are going to be looking at lactose fermentation. But there is a secondary substrate in HEA, and that is sodium thiosulfate. Now, we will talk about that after we talk about the lactose fermentation part. But that is another substrate that's in the media. Now, there's other stuff that's there too, but we're not going to worry about that. We're just going to talk about the lactose and the sodium thiosulfate. So the selective agent in HEA is bile salts. So again, we're going to be selecting for gram-negative organisms. So gram-positives are not going to grow on this because we are looking for enteric bacteria, which are gram-negatives, most of them. So the bile salt selects against gram-positives, but HEA is different than the others because E. coli is not going to grow on HEA. Because for HEA, we are looking for different enteric pathogens other than E. coli, like Salmonella and Shigella. So E. coli, we aren't looking for it anymore. So HEA actually selects against E. coli, even though it is a gram-negative organism. It selects against it. So E. coli is not going to grow on HEA. That's the only gram-negative organism that's not going to grow on HEA. So see, I told you it gets a little complicated. So selecting for gram-negatives except for E. coli, and our indicator is bromothymol blue. Now, this one looks a lot different than the other pH indicators that we've talked about because for McConkey, McConkey, ENV, and MSA, we were looking for fermenters. So our positive results were coming from fermenters. For HEA, my positive results are non-fermenters. So it's different than ENB and McConkey. For ENB and McConkey both, we were looking for E. coli. Well, E. coli is a lactose fermenter, so that's my positive results. But for HEA, we're not looking for that anymore. We're looking for Salmonella and Shigella. Both of those are non-fermenters. So our positive results are going to be non-fermenters. Well, if they're non-fermenters, they're going to be producing neutral or basic end products. Bromothymol blue turns blue, well, not here, it's a blue-green color. But for a positive result, you're getting a blue-green color because you're looking for neutral or basic end products. The media starts out green and turns a blue-green for a positive result. So a positive result for HEA is a non-fermenter, and it is a blue-green color. The agar and the colonies turn blue-green. For a negative result for HEA, so that means that the bacteria fermented the lactose, you get orange/yellow colonies, and the agar also turns color. It's like a bright coral color, and you'll see that whenever I look at read the results. So for a positive test for HEA, number one, you're going to get growth. It's going to grow. Number two, you're going to get blue-green colonies. For a negative result, it grew, but it gives you bright coral color colonies. That means that this bacteria was a gram-negative organism, but it's not Salmonella or Shigella because it fermented the lactose. So if you're not confused yet, let's continue on with the secondary substrate, which is sodium thiosulfate. Okay? So there's sodium thiosulfate in the media. Some bacteria can take the sodium thiosulfate and turn it into hydrogen sulfide, which is this right here, H2S. Some can, some can't. The H2S reacts with iron that's in the media and turns into iron sulfide. This produces black colonies. So the colonies will be blue-green, but they will have a black center to them. So you will have growth, which is gram-negative. You will have blue-green colonies, and then they will have a black center. If it's positive for hydrogen sulfide production. So the reason why we do this, Salmonella, of course, produces blue-green colonies. It grows, produces blue-green colonies, but it gives us black centers to the blue-green colonies. So that would be indicative of Salmonella. Shigella, on the other hand, which is a completely different type of bacteria, gives us blue-green colonies, but does not have black centers. Okay? And so, of course, salmonella comes from chicken. Pretty much everybody knows about salmonella food poisoning. If you eat raw chicken or undercooked chicken, or if you don't wash your hands properly whenever you're dealing with it, you can get salmonella food poisoning. Shigella is a little less known. That's because it's not really around here. You get it from drinking contaminated water, usually from Mexico. So this causes dysentery. So it's a little bit different. It's still food poisoning, but it's just a different form from salmonella. And it's still coming, it's an enteric bacteria, so it's coming from feces contaminated, drinking feces contaminated water, okay? So that's the reason why we look at the H2S production. If H2S is produced, you're going to get the black colonies, okay? Now, for HEA, I'm going to be inoculating two plates for two different kinds of bacteria. I'm going to be using Citrobacter frundi, which is a gram-negative organism. It is a fermenter, so it's going to give me that coral color, but also it produces H2S. So hopefully we'll get those black colonies on the plate, so you can see that. And then Pseudomonas is, of course, a gram-negative non-fermenter. So it's going to give me the blue-green colonies that I'm looking for a positive test for HEA. Okay, so now I'm going to go ahead and inoculate my plates, and these are all normal streak plates. So no swabbing or anything like that, no going straight down the center. These are all going to be normal streak plates. So now we're going to go ahead and do that, and then we'll be finished with our video. I've got all my plates here. I've already labeled all of them with, of course, the bacterial name and the type of media that they are. I don't usually have to label the media just because I know what it is, but if you were doing this in lab, you would need to make sure that you label what kind of media it is so you don't get confused. And I've got all of my bacteria, and actually I'm going to be using from my original plates today, just because it usually gives better results because there's more bacteria that's placed on the plate using solid media. So I'm going to start out with the PEA. Here's what the PEA looks like starting out. Okay, so it's a clear plate, really no color to it. Okay, it's kind of like a yellowy looking color a little bit, but that's what it starts out as. So my two bacteria that I'm using for this are going to be Bacillus and E. coli. All right, so I'm using 2 loops just because it goes by a lot faster whenever you use 2 loops. So I'll flame my loops. And like I said, these are going to be normal streak plates. One off. That's not good. Alright, so I'm going to finish my streak plate. Okay, there's one. That was my Bacillus. Now I'm going to do my E. coli, which of course for PEA should not grow because it's a gram-negative organism. And if you notice, I don't reflame my loop between 2 and 3 because I simply flip my loop over. If you flip the loop over, then you're using a new fresh side so that doesn't have any bacteria on it. Alright, so my 2 PEA plates are done, so I'm going to add those to my box. Now I'm going to move on to the McConkey. Here's what the McConkey looks like before. Okay, it's like a pinkish color. Okay, because remember that neutral red turns that hot pink color. So, and the 2 bacteria I'm using for this are E. coli and Pseudomonas. So I'm going to do my E. coli plate first. Done. Now I'm going to do my Pseudomonas plate. Okay, the conky plates are done. Add those to my box. Now I'm going to do the EMB plates. So here's what the EMB looks like. It's kind of like a purpley, weird looking color. Starts out as. So for these, I'm going to be using E. coli, Enterobacter, and Pseudomonas. Those stacked up. First, I'm going to do the E. coli. Okay. That's done. Now I'm going to do my Enterobacter, and remember Enterobacter is going to give me growth, and then it's just going to give me regular purple colonies. So it's a lactose fermenter and it's gram-negative. Okay. Now, the last one is the Pseudomonas. If you remember, Pseudomonas is the non-fermenter. So, it's gram-negative, so it is going to grow, but it's not going to give me those purple colonies. They're going to be colorless. Now, if you notice, the media is already kind of like a purple color. So that's why I say the same color as the media, because a lot of times people will look at it and say, well, the colonies look purple to me. Well, they look purple because the media already kind of has a purple color to it. So, and when you see how purple the E. coli and the Enterobacter is going to be, you're going to understand what I mean. All right, here's last. This is the HEA. So if you see, it is a green color already. That's that bromothymol blue that's in there, giving me that green color. So I'm going to do Citrobacter frundi and Pseudomonas for these two. So I'm going to do the Citrobacter frundi first. This is the one that is a gram-negative lactose fermenter. So it should give me the negative result for HEA, which is the coral color. But it does produce H2S, so hopefully we'll get some black colonies. Sometimes it produces black colonies and sometimes it doesn't. It can be kind of temperamental sometimes. This media is soft. Okay. And lastly, my Pseudomonas. All right, so now we're completely finished with inoculating all of our plates. Hey everybody, we are back here today to read our results for our selective and differential media. We let them incubate for 24 hours, and if you remember, we had 4 different kinds of media. We have PEA, McConkey, EMB, and HEA. So the first plate that we are going to look at together today is going to be the PEA plate. And if you remember, in the last video, I told you that I group PEA by itself because it's the only one that is strictly selective. So I like to read it first because it's different than the other 3. So let's continue on with reading the PEA. And if you see, I've got the results up on the board, what it should look like. So let's read PEA. So I've got both of them right here. Okay, so if you remember, I did a streak plate with both of these in the last video. So here's E. coli and here's Bacillus. So you'll notice that there is absolutely no growth on the E. coli plate. Nothing there. All you see are my streak marks. So that tells me that E. coli is a gram-negative organism. Remember, PEA is selective for gram-positives. So only gram-positive organisms are going to grow. So E. coli is gram-negative, no growth. Bacillus, however, is a gram-positive organism. So I've got lots of beautiful growth on my plate. Look at that streak plate. It's gorgeous. So that tells me that Bacillus is a gram-positive organism. Now, since it is strictly selective, that's the only information that I'm going to get from PEA. You streak it on the plate, it grows, that tells me it's a gram-positive organism. You streak it and it doesn't grow, that tells me it's a gram-negative organism. And that's all that it's going to tell you. So, very easy, very straightforward for PEA. Now, moving on to the next three. Remember, these are the ones that are both selective and differential. And they're all very similar, but a little bit different. So they're all selecting for gram-negatives. So only gram-negatives are going to grow. And the substrate for all of these is lactose. And we are looking at fermentation of that lactose. So can the bacteria ferment the lactose, yes or no? And that's the question that we're asking. So we're going to start out with the McConkey plate. Okay, so here's my McConkey plate. So the McConkey, again, remember we're specifically looking for E. coli for these. Or a gram-negative lactose fermenter is what we're looking for. So first of all, it has to be a gram-negative organism for it to grow. So if you see, both of my McConkey plates have growth on them. So both of these organisms are gram-negatives that I streaked on my McConkey plate. And then second half of it, which is the differential, is the fermentation part. So can the bacteria ferment the lactose, yes or no? Now, you might have a bacteria that is a fermenter, but can't ferment the lactose. If it can ferment other sugars, it can definitely do that. But we're specifically looking for a lactose fermenter. So the two bacteria that I have here are Pseudomonas and E. coli. So if you look at the board, a positive result is a hot pink colony. It says red in your book, but like I said, it's going to look hot pink. And that is positive for lactose fermentation. That means this bacteria can ferment the lactose. And if you see, here's my E. coli, and look at those bright, hot pink colonies. I told you, it says red in your lab manual, but it's more like a hot pink. So this is a positive result for McConkey. This bacteria is a gram-negative lactose fermenter. Now, there are other gram-negative lactose fermenters. E. coli is not the only one, but that's what we use in this lab because it gives such great results. Now, the other bacteria I looked at was Pseudomonas. So a negative reaction would be colorless colonies. So as you can see, they are the same color as the media pretty much. Okay? So they're very colorless. There's really no color to these. They're pretty much clear. So that's a negative reaction for McConkey. Okay? There's a huge difference between these two. Okay? So all these colonies have that bright hot pink color to them. That's positive. These are colorless. They're clear. Okay? So this is a negative reaction for McConkey. This means Pseudomonas is gram-negative. It grew. But it cannot ferment lactose. So you get the colorless colonies. Okay, so that's it for McConkey. So now we're going to move on to the EMB. Now, if you remember EMB, we are again looking for gram-negative lactose fermenters, and specifically EMB is used to identify E. coli among other enteric bacteria because E. coli does something special on EMB. It gives you that bright green metallic sheen on top of the purple colonies. So it's E. coli is gram-negative, it's going to grow. It's a lactose fermenter, you're going to have purple colonies. And then on top of those purple colonies, you're going to get a bright green sheen. Now, if the organism is not E. coli, but it's still a gram-negative lactose fermenter, then you're going to get growth and the colonies are going to be purple. And if it's a gram-negative organism, but it's not a lactose fermenter, then you are going to get growth, but the colonies are going to be colorless. Okay, so let's look at a negative result first. So we're going to look at Pseudomonas. Okay, so as you can see for Pseudomonas, I have growth there. Okay, not a whole lot, but I've got some growth there, and they are colorless colonies. Okay, so you can see they're kind of hard to see, but they are colorless colonies. So Pseudomonas is gram-negative, but it is not a lactose fermenter. Now, let's look at Enterobacter. Enterobacter is gram-negative, and it is a lactose fermenter, but it's not E. coli. So let's look at that result. So here's that. You see the purple lavender color? Okay. If I were to let these grow a little bit longer, the colonies would get a little bit bigger, but you can still see that purple color. So it is a gram-negative lactose fermenter. Now here is my favorite one, which is E. coli. So I should get purple colonies with the bright green sheen on top. And wow, look at that. Isn't that beautiful? It is a beautiful, bright green sheen. And whenever you see that, you know that that is E. coli. Look at the difference. Look at that difference. This is the purple. This is the E. coli. It's very in your face, bright green sheen. So that's my E. coli, my positives for E and B. Beautiful, bright green sheen. So if you were to see an E and B plate and it has the purple colonies and this green sheen, you immediately know that that's going to be E. coli or a strain of E. coli. That's what's so cool about E and B. Now, the last set of media that we are going to look at is the HEA. And remember, this one's a little bit more complicated because even though the substrate is lactose, we are looking for non-lactose fermenters. Okay, if you remember from the last video, I told you these other two, macaque and E and B, we were looking for gram-negative lactose fermenters. That's the kind of bacteria, the pathogens that we were looking for on those plates or trying to identify on those plates. For HEA, we're trying to identify non-lactose fermenters like Salmonella and Shigella. Now, Salmonella and Shigella, they can ferment other sugars, but they can't ferment lactose. So that's why we have lactose as the substrate in the HEA. So remember, it selects 4 gram-negatives except for E. coli. Remember I told you, we're not worried about E. coli here. So E. coli is not going to grow on HEA. So for the HEA results, we didn't, for those plates, I did not streak in E. coli because it's not going to grow. So let's look at the first part, which is the lactose fermentation part. So a positive result for HEA is a blue-green color, and that means that it is a non-lactose fermenter. So a positive result is a coral color, orange/yellow or bright coral. That's a negative result. That means that that bacteria is a lactose fermenter, and we're not interested in that whenever we're looking at the HEA plates. So let's look at our two. So we'll start out with a negative result for HEA, which is a coral color. Dun-dun-dun. Look at that coral color. Those colonies are bright coral. And remember, this is Citrobacter frundi. So it is a gram-negative organism, and it is a lactose fermenter. Bright coral color. So this is not Salmonella or Shigella. Okay? It is a negative result for HEA. Now let's look at a positive result for HEA. Pseudomonas. There's my blue-green. Pseudomonas is a gram-negative, non-lactose fermenter. So it's going to give me that positive result for HEA. Nice, beautiful blue-green colonies. All right, so that's the first part of the HEA results. The second part of the HEA results is looking for hydrogen sulfide production. Remember I told you some bacteria can take the sodium thiosulfate that's in the media and produce H2S. H2S, when it's produced, reacts with iron in the media and produces iron sulfide, and that produces black colonies. So You can have a blue-green plate with black colonies, a blue-green plate with no black colonies, but you can also have a negative result with black colonies. I know this is the positive and negative for the H2S, but you can also have a coral plate with black colonies. That just means that bacteria is a gram-negative lactose fermenter that produces H2S. So that's what we have today. On the Citrobacter frundi plate, you will see right there, in that deep area right there, you will see that black color, okay? That is H2S that has been produced, and then you get that iron sulfide that's producing the black color. Also, in this area here, you see a little bit of the black, okay? It's most indicative right in that deep spot right there, okay? So, and like I said, sometimes you get the black, sometimes you don't. This one we did get some black, so that's good. That's what we want to see. If I were to let this go a little bit longer, you might get a little bit more black, but I wanted to go ahead and make the video for y'all and get it posted. So this would be a negative result for HEA. This one would be a positive reaction for HEA, non-lactose fermenter, with no H2S production because there's no black on that plate. Okay, so coral color, lactose fermenter, blue-green color, non-lactose fermenter for HEA. Okay, so that's it for these results for your selective and differential media. I know it's lots of colors, lots of things to remember, but if you try and group these in your mind into groups that are alike, I know that always helps me. And try not to just memorize colors. Try to memorize the pH indicators, and then that will help you to remember what colors things are turning. Okay, phenol red, neutral red, and in the next video that I'm going to post, you're going to have even more colors that you're going to be looking at. So if you just memorize colors, you're probably going to get confused. So please don't do that. Please make sure that you go through and memorize that whole chart, and to help you know what colors things are going to be, and know which pH indicators and why they're changing the colors that they're changing. It will make more sense if you actually learn why they're changing the colors that they're changing.